Okay, today we're going to be talking about mutations. So, a mutation is a mistake in a DNA's organism. Okay? So that's your first blank. It is a mistake in a DNA's organism, and these are permanent changes. This is not something that once it's done, you cannot go back and fix it. It's a permanent change in the DNA. All right, so types of mutations. There are two types of mutations that you need to know about. Okay? The first one is a point mutation, and the second kind is frame shift mutation. Point mutations and frame shift mutations. Oops. Oh my gosh. All right, a point mutation. A point mutation occurs when a nucleotide is accidentally replaced by a different nucleotide. A nucleotide is replaced by a different nucleotide or a different letter. So in this picture, okay, this DNA is supposed to read, this DNA is supposed to read CTT. Okay, but this person has a mutation, so instead their DNA reads CAT. All right, it's just a change of one letter. That's a point mutation. Okay, so here we can see, right, one nucleotide is accidentally replaced by another nucleotide. Therefore, our mRNA is supposed to read GAA. Now, if you flip over your worksheet that goes with this lecture, you will see that there is a codon chart for you to read. Okay, if you look at the back, there's a codon chart. What does GAA code for on your codon chart? Good, glutamic acid. The amino acid that should be coded for is glutamic acid. However, this person has a mutation in their DNA, and it changed that T to an A. So now their mRNA reads GUA. What does that code for on the codon chart? Valine. That difference right there is a huge difference. All right? It may not seem like it. Like, who cares? It's just one amino acid in the protein chain. Okay? However, that one change in amino acid causes a pathology or a disease. Okay? Just by changing that one amino acid, you change the entire protein that determines the shape of red blood cells. Okay? This causes the red blood cells to be boomerang-shaped or sickle-shaped. A sickle is the thing that the Grim Reaper holds. Y'all know what I'm talking about with the Grim Reaper? No. Okay, remember Grim Reaper? Let's see if I can draw him. Okay, here's the Grim Reaper. He's a sad person, right? And he holds this thing. Yeah. You know what I'm talking about now? Yeah. Okay, this part is called a sickle. See how this red blood cell is sickle-shaped? Yeah. That is called sickle cell anemia. All right? It is a disease in which red blood cells, some of them have normal shapes, some of them have this sickle shape. And it can cause a variety of health problems. But we will come back to sickle cell anemia when we talk about evolution, because there's actually some benefits to having sickle cell anemia. okay, which is why we've seen it stay around in the population for so long. So from changing just that one letter in the DNA, these people have a disease called sickle cell anemia. So we can see that that one mutation can make a big difference, right? However, sometimes, even though the nucleotide changes, it still codes for the same amino acid. Okay, so looking at your codon chart, all right, we're going to look at this piece of DNA. Here's the piece we're focusing on. The DNA was supposed to say AAA. Our mutated DNA says AAG. Did everyone see that?
What it's supposed to say. So wild type is the regular DNA?
Yes, wild type is how it's supposed to be, and then the mutated one is the one with the mutation. So, very good. So normally AAA gives us an RNA of UUU. If you look at your codon chart, what does UUU code for? Phenylalanine. Phenylalanine. Okay? PHE. In the mutation, now our RNA reads UUC. Look at your codon chart. What does UUC code for? It still codes for phenylalanine. It codes for the same thing. This, you know how on your codon chart, there's a couple things that all code for the same thing. That's another way our bodies protect ourselves from having problems with mutations. Even though mutations are incredibly rare, the redundancy built into the codon chart protects us from mutations always being a bad thing or always being a noticeable thing. So we call these silent mutations because you don't see them. They're not noticeable, right? Like the K in knife is silent. You don't know it's there unless you spell it out. You don't say it. Okay? So, a silent mutation does not change the protein that gets made. The final protein still comes out exactly the same, and it does not affect the phenotype of the organism. Your phenotype is what you look like. All right, second type of mutations, type 2. These are called frameshift mutations. A frameshift mutation is when nucleotides accidentally become inserted or deleted. So all of a sudden your DNA is like, you know what'd be fun? Let's put an extra A in here. Or, oops, there was a G there. Our bad. We forgot it. So this changes the reading frame of your DNA. How many letters do we read at a time in DNA or RNA? 3, right? AUG, CCG. That's how you read your codon chart, right? Nod your head if that makes sense. Fabulous. So if we have a frameshift mutation, we can think of the original piece of DNA like a sentence. It's all 3-letter words, right? The red cat sat said. That's sadly the most creative sentence. I apologize that sentence is so lame. But that's like our DNA, all right? Now with a frameshift mutation, if we accidentally delete a letter, then let's say we delete H, then our whole sentence changes if we're still reading it every 3 letters. Now it says tur, ed, k, ats, ad. Does that make sense anymore? No. So if you delete a letter out of your DNA and you start trying to read it again, it's not going to be the same thing, right? It's going to mess up everything down the line from there. So the same thing happens if you add a letter, right? All of a sudden you add in an extra H. Every 3 letters becomes a new combination instead of what it should be. Whoops. Oh, okay, so frame shift mutation, here's another example using DNA letters or RNA letters. ATG, GAA, GCA, CGT is how it's supposed to go, right? We accidentally said, oops, I forgot about that guanine, and now it reads ATG, so it still codes for methionine to start. But then the next series reads what? AAG. Does AAG code for the same thing? No. So now you can see the rest of the DNA and the rest of the protein is going to be all messed up. Does that part make sense to you guys?
Yeah.
Excellent.
Wait, if you take out
All right, so mutations. Mutations are everywhere. They are all over the place, okay? All types of genetic diversity were caused by mutation at some point. For example, blue eyes are a mutation of brown eyes. Mutations can result in a harmful alteration to the organism, so it could cause a disease or a pathology. It could cause no difference, right? We saw that in the silent mutation. It made absolutely no difference. Or it could even be a benefit. It might make you like Superman. Rarely does it, but it might. You never know. So it could be a benefit to the organism. And truly, mutations are what drive change in a species. That's the only reason, really, that we all look so different, is different mutations over time being added into our gene pool. All right? And with that being said, I have these 2 videos. We're going to watch the top one this morning, and I'll save the other one for tomorrow morning. But these are 2 pathologies or diseases that are caused by genetic mutations. All right? Then after this, I will explain the worksheet to you, and we'll go on from there. Sound good?